All right, thank you. My name also is Corie, not to be confused with the supercomputer at NERSC. All right, let's just see here. Okay. All right, so thanks for allowing me to talk to you about the Advanced Light Source, which is a synchrotron facility. And first, to just tell you where we are, we're very close by. We're actually right across the bay in beautiful Berkeley. And if you look at this picture here, this is the Advanced Light Source, and we are sitting approximately here right now in San Francisco, a short jaunt across the perpetually clogged Bay Bridge. We're probably also about an hour away, depending on the time of day. So the Advanced Light Source is a machine dedicated to producing very high brightness X-rays in both the soft and the hard X-ray regimes. And when I think about what you can use X-rays for, you can do several things. So you can shoot X-rays at a sample and see what gets absorbed. You can shoot X-rays at a sample and see what gets transmitted. You can see what fluoresces as a result of shooting X-rays at a sample. You can see what kind of electrons are produced when you shoot X-rays at a sample. Or you can look at diffraction. And all these different techniques give you different types of information about the sample that you're looking at. So X-rays are really a really good characterization technique. And so the stations where these X-rays are produced are called beamlines. This facility has 45 beamlines, all operating simultaneously pretty much year-round, except for a month or so a year in which we have maintenance periods. This is a DOE facility open to scientists from all across the country and in fact all across the world. We have about 2,000 users per year at this facility. So I imagine that probably what most people in this audience are interested in is what are the biological beamlines at the ALS. So here's the map of biology techniques that can be done at the ALS. As you can see, it's a pretty significant portion of the beamlines at the ALS. So we have diffraction techniques. We of course have crystallography. We have tomography in both the hard and the soft X-rays. We have spectroscopy beamlines. We have small angle scattering, one small angle scattering beamline. And we have an X-ray footprinting beamline, which I don't have time to talk about, but it's a technique that's near and dear to my heart, and I'm happy to tell you more about it if you want to ask me about it after this talk. So how does a typical experiment work? You basically apply for time through the ALS user's office. Once you have beam time assigned to you. So for your proposal will get vetted and scored, and then hopefully you get allocated beam time. Once you're allocated beam time, you can either just send samples or you can contact the beamline scientist. If you haven't used the method before, you can contact the beamline scientist who will tell you how to prepare your samples and how to collect your data. We have users who come on site. We have a significant portion of the crystallography users who now run remotely. Something like 80% of our users are remote for crystallography. And the shifts for crystallography tend to be on the order of 4 or 8 hours, where some of the physics experiments done at the ALS tend to be on the order of weeks. So it's interesting to me that some of the crystallography users who are remote, who are so experienced, we actually never see them anymore. So they apply for time and they get time, they send their samples, and they never interact with us. On the other hand, we have users who come and are very new and need a lot of help and need to learn how to prepare their samples. So we interact with people on all those levels. And so this is accessible to users to be on site as well. So we offer biochemistry and chemistry labs. We have materials for use. And of course, we're always open to collaboration with users. So in table form, we have the infrared beamlines. We have the multi-wavelength crystallography beamlines. We have single wavelength crystallography beamlines. We have small angle scattering beamline. And these are the different energy ranges. And it's always kind of interesting to me, too, that we really span through these techniques a really large range of length scales. So from the order of millimeters or centimeters, looking at imaging of whole animals, down to the atomic level resolution of a crystal structure. And I did want to point out as well that we have a significant portion of our users who are from the pharmaceutical industry, and they're using crystallography for structure-based drug design. So if you want to learn more about it, go to als.lbl.gov, and you can sign in, get yourself an account, pretty easily here. And then I encourage you also to go to the beamlines tab where you can learn about the different beamlines and find out who the contacts are for the various beamlines at the ALS. And just to close, this is a DOE-funded facility funded by the DOE BES, but I wanted to point out that we also have staff at the ALS who have some funding from the NIH and from the Howard Hughes Medical Institute and from pharmaceutical industry. And thank you for your attention.